Modern affinity reagents, recombinant antibodies, and aptamers aims to familiarize antibody users with the state of the science of recombinant antibodies and aptamers, how they can be incorporated into the protocols that require affinity reagents, and how to gain momentum in the transition to these scientifically superior non-animal reagents. There's a growing concern about the reproducibility of animal-derived monoclonal antibodies, which contributes to the lack of reproducibility of biomedical research. Recent publications cite documented evidence of the lack of affinity, specificity, and reproducibility of animal-derived antibodies, and describe how their use has wasted tremendous amounts of money, time, and experimental samples. The need for higher quality affinity reagents has prompted the development of methods that provide scientific, economic, and time-saving advantages and do not require the use of animals. Two non-animal technologies that can replace the use of animal-derived monoclonal antibodies are recombinant antibodies and aptamers. These technologies can be used in the same applications as monoclonal antibodies produced in animals, including in basic research, regulatory testing, and clinical applications. Recombinant antibodies are protein-based reagents that are selected from libraries of genes encoding slightly different antibody proteins for their affinity to bind to target antigens. Aptamers are short, single-stranded DNA or RNA oligonucleotides and are similar to antibodies in that they can detect and characterize their targets and modify the activity of their targets. The process of screening large nucleic acid libraries to develop aptamers, called Celix, and its modifications are described in the review. Advantages of recombinant antibodies and aptamers include they can be produced more rapidly and with higher affinity levels and binding specificity. They have greater versatility, allowing, for example, the ability to generate antibodies and aptamers against toxic or non-immunogenic antigens. Batch-to-batch variation is limited because the sequences are known and because their generation does not rely on biological processes. And hundreds of thousands of animals are used in the production of affinity reagents every year, but recombinant antibody and aptamer development do not require animal use. Specifics about these and additional advantages, as well as considerations of these technologies, are discussed in the review. A number of recommendations to assist researchers in the transition from animal-derived antibodies to non-animal sources are provided in the review, including a consortium of industry, academia, government, and non-governmental organizations should fund the development of commonly used affinity reagents to be made available to all researchers. University and federal funding incentives should be made available for researchers, and researchers should become familiar with and use non-animal research methods whenever possible. Companies offering recombinant antibodies and aptamers are listed in the review. From a scientific perspective, it is clear that there is a strong need for more reliable, specific, and versatile affinity reagents. Once technological knowledge and the supporting infrastructure are established, recombinant antibodies and aptamers require less time to produce, require less purified antigen, and can be created against a larger number of targets than monoclonal antibodies derived from animals. These technologies are the way forward to increase scientific validity and reproducibility and to accelerated research in the life sciences.